Here and yeah, get started.
Thank you for inviting me here. So this is probably going to be the most far out talk here. I'm hoping it's not, or I'm dreading that it is the most far out talk. And basically, I'm going to try to say something about how this line of inquiry might have something to do with what most of you are doing. So I'm going to start talking about brain emulation. What is emulation? Well, normally when we say that something emulates something else, we mean that it's kind of fake. I might try to emulate Danish, but all Danes going to notice that it's totally a Swede trying to speak Danish. It's just going to be very fake. There is some similarity, but that's about it. But in computer science, we use it in a slightly more specific way. That was my first computer, a Sinclair ZX81 computer, 1 kilobyte of memory, 44 times 63 in pixels, black and white resolution when you connected it to your television set. It's, of course, very dear to my heart. It's also, yeah, it's hard to find a television set. But I can run a piece of software, of course, on my modern PC that actually does a one-to-one simulation of that computer. Indeed, this is not just that it produces the same output. It actually simulates the states of a processor, the states of electronics, to a fairly high resolution. That emulator even allows you to jostle the computer, which generally causes a crash, because there is a mechanical issue with the data bus. It's literally that level of emulation. And this is, of course, useful if you have dear old computers that you might want to run. So the 6502 microprocessor also used to be a very loved one. It was important for a lot of old computer games and old computers. And it turns out that the schematics at Motorola actually, or was it Intel, I forgot, actually are not accurate. It turns out that the actual photographic masks used to make the chip are different from the actual circuit diagrams. There was some debugging done manually that was never documented. But this project, they took a processor and imaged it under a microscope, and then they basically used computer vision methods to discern what the transistors are and deduce the actual connectivity, the actual functions, etc. And it turns out that then you can run a piece of software that emulates it accurately, and you can actually run Space Invaders in original gorgeous speed and color. So the interesting idea here is that you have a system, you scan it, maybe in a destructive way. That processor was kind of destroyed by the scanning because they had to literally open up the capsule for it. And then you generate a computer model that is a one-to-one image. Now, we might imagine doing the same thing for a brain. This is basically the idea with brain emulation. You start out with an actual biological brain, and then this is, of course, where you can start arguing about which methodology might be used. But one possibility might be you freeze it or fix it, and then you start slicing it up and image it using electron microscopy. You end up with a three-dimensional model of what's where. You then generate a computational model, run using the normal Hodgkin-Huxley equations and other biophysics. And if everything has gone well and you have a sufficiently big computer and a body model and hand wave, you end up with an organism that behaves the same as the original owner of the brain. So this sometimes called mind uploading. I hate that term because we actually don't really know what the mind is. And uploading also seems to imply that there is a trivial relationship between the information in a brain and what you end up in a computer. And it's way more intricate. It's not just like moving a file from one computer to another. There is a fair bit of processing. But at the same time, there is an interesting question. Can this actually be done? So back in 2008, we at the Future Humanity Institute, we had a little workshop over at St. Hilda's College in Oxford, where we ended up developing this roadmap. It's a technical report. It's kind of describing state of the art at the time, what we were thinking was necessary, where we knew there was enough technology or not enough, including questions that needed to be resolved. And there is a fairly large set of assumptions you need to believe in order to think that brain emulation could actually work. You could say that, well, actually, maybe there are non-physical parts of the brain that are really important that can never be scanned. There is an important question about, well, is it computable? I think most people in biology and neuroscience would say, wait a minute, uncomputable processes are really, really weird ones. It's only physicists like Roger Penrose who believe that there is actual uncomputable stuff going on. And even if it's not uncomputable, but he's just right about quantum mechanics mattering in the brain, okay, we're still in trouble with the project because it's rather hard to scan quantum states. But practically nobody believes that kind of quantum biology is a major thing in neuroscience. This might be a way of finding out the hard way. There is also an interesting aspect that this whole idea is based on that if we can understand the parts of a system well enough and put them together in the right way, then you would get the full system. So my standard metaphor as a Swede is, of course, talking about assembling IKEA furniture. Over here, of course, we should be talking about Lego. And the basic thing is, if you have the right parts and you have a blueprint of exactly where they go, you can make a structure, even though you don't understand the functions. I don't understand the carpentry going into making a good bookshelf, but I know how to assemble an IKEA Billy bookshelf fairly well, and it produces a workable bookshelf. And we might imagine that the same thing might apply to brains. This is, again, an interesting thing that gets some philosophers very worked up. And it's also a bit different from what many of you do, because this is not normal science. In normal science, we take a system, we try to pick it apart and model it in order to understand it. We want a high-level understanding of what is going on. What does the mTOR1 pathway actually do? What happens if we do certain things to it? We want to have a predictive model in the sense that we have a high-level understanding. We don't want a predictive model in the sense that we have a computer program that we can manipulate and totally get the right output from, but can never understand. Brain emulation aims at making one of these less understandable systems. So again, a lot of neuroscientists are kind of getting, what? Why should we even care about this? This looks like nonsense. But one obvious reason we might want that is, of course, if you can make a small or a big neural system work, you can actually do a lot of experiments on them. You can actually get the understanding in a more easy way. This is, of course, how we computational neuroscientists typically work on our systems, although most of them tend to be smaller and more well-defined. There are other interesting issues here, like scale separation. Is it that the microscale on a sufficiently fine resolution can be averaged away so we can ignore it? Or is it that everything interacts with everything across scales? So this is a little bit like the issue of statistical mechanics. In a gas, the collisions between molecules average out very nicely, so we get macro states like pressure and temperature that make sense. This is great, because that means that as an engineer, you can totally ignore those molecules in many applications and just deal with temperature and pressure. Unfortunately, in fluid dynamics, in turbulent flows, big whirlpools generate small whirlpools all the way down to a very fine scale. So if you want to simulate fluid dynamics, you actually need to take a lot of scales into account. And indeed, there might not be any proper scale separation, which means that if you leave out the finest, smaller scales, you actually get what's called dry water by the computational fluid dynamics people. You get something that behaves almost like water, but not quite. Now, are brains like turbulent water or air? We don't know. This is actually a really cool science question I'm hoping to work on more with some other neuroscientists. There are other issues, of course, about can we actually simulate the components? I think many people are optimistic about that. But again, people will always bring up exotica. What about electromagnetic interactions? What about body temperature? Temperature affects neural function quite a bit. It might be that there is a fairly long list of stuff you need to take into account here. Some of it, I think, is pretty straightforward, like temperature. The electric fields would be annoying because now you need to have an EM solver connected to your biophysics model. But it's not terribly impossible because we have EM solvers. There are other interactions that might be harder to deal with. Heaven forbid that we have quantum stuff going on in our heads. In that case, this is going to be much harder. And then, of course, there is the issue that brains in themselves don't work very well. They need bodies. When I look around the room, it's not just that my brain is getting signals from the eyes. I'm moving my eyes. I'm performing saccades all the time, focusing on different things. The brain is doing a lot of very elaborate balancing when I'm moving my eyes around. The physics, if you leave out the physics of your eye simulation, you're probably going to get a rather confused and nauseous brain because it doesn't function well. You need to have some body simulation. That might not have to be a body emulation. It's probably unlikely that you need to simulate the interior of bones other than their mass and the moment of inertia. But it's very likely that there is a fair bit of information about the body model you need to get in going here. Now, since 2008, a lot of stuff has been happening. I'm going to review some of it. So one of the things we never expected was that biotech would be so awesome. We were mostly thinking microscopy, and people are coming up with all sorts of really intriguing things like molecular ticker tape, where you make cells that document their activity in the form of long proteins, which you then can extract. Or DNA barcoding, not just for organisms, but for individual neurons, and maybe even for individual synapses. And the expansion microscopy, which is I'm going to return to. Also, of course, there is a lot of stuff happening in neurotech. You have very large simulations that by now are brain scale. The problem is many of these large simulations are based on some model of a brain rather than an actual brain. So we have some pretty impressive data sets. Also, of course, we have some contentful simulation like Spawn, which is a model, but it's still kind of big as many small brains. People have also been doing a lot of research, and there is an interesting shift in Moore's law. When we were doing our workshop, that was just at the cusp when microprocessors were starting to slow down. They previously had been increasing the number of hertz they're running on, and now instead you can't get them much faster in terms of frequency. People started using more cores. And the GPU revolution has happened in the intervening, which is totally fine for most of these applications, fortunately for us, because parallel processing works rather well with brains. One interesting issue is that we were very bullish about, of course, we're going to try to upload C. elegans, that nematode worm. After all, we've known its connectome since the 1980s. How hard can it be with 303 neurons? Turns out that it's darn hard to do this well because it's so tiny, because you need to find the synaptic weights between the neurons. And getting glass electrodes that tiny into an organism which has an integument under pressure, that turned out to be way harder than expected. If we had focused on the pond snail instead, we would probably be much more successful. And of course, since then, a lot of AI applications have shown up too, which is probably going to accelerate this process quite a bit. So I mentioned expansion microscopy a bit earlier. To me, this is mind-blowing because it's also a practical thing, and I'm a theory guy, so I never really trust the practical stuff. You take your tissue sample, you embed it in a gel matrix, you make the matrix expand, and now you have a bigger thing to look at, which means that those things that you might have wanted to study previously under an electron microscope, but that would be very hard because electron microscopes are expensive and finicky things, and it might be that you also want to see chemical things that are hard to study under an electron microscope. Now you can use an optical microscope with many of the very cool modalities clever microscopists have developed over the years. So this way you can literally make pyramidal cells large enough to be seen by the naked eye if you expand them far enough. It still has various limitations, but people are getting more and more optimistic about seeing nanoscale resolution details and actually building up rich models of entire brains and the connectomes. Now, one of the fundamental problems is, of course, a ridiculous amount of data. Back in 2008, we were kind of dreaming about making a cortical column observatory. We didn't even believe that we could store that much data in a computer, so maybe some kind of tapes with fixated tissue on that could be moved in front of microscopes. So when you're moving around and trying to do your 3D reconstruction, you would be literally moving tapes of tissue around. Turns out that, we're getting to the era where we get full connectomes and can actually store them in the cloud. So right now the star is, of course, the fruit fly. Who doesn't love a fruit fly? Especially one with a colorful brain like this. So by now, we have actually what looks like the full fruit fly connectome. And that's nice. You can do quite a lot of interesting science on this already. People, as I will show, have been doing very funky stuff. But as I said, we were dreaming about doing this cortical column, but that was kind of this remote observatory idea. And then this weird feeling I had at a conference sitting right next to one of the guys on this paper. Oh yes, we scanned a cubic millimeter of actual human cortex at the electron microscope resolution. And yeah, you can go onto their site too and look at it. And there is a lot of weird stuff in there. So just from a pure science perspective, figuring out what the heck some of those things are. Why do those axons connect in that crazy way? There is barber pole axons that wind around dendrites. Nobody has ever expected that. We neuroscientists have no clue what that would be good for, but they exist. So this is a very rich data set. It's slightly unique, of course. It's rather rare that when you can get hold of a good brain tissue and then scan it like this. And this was expensive to do, but it shows what we currently have. And this is 16 times bigger than that cortical column we were dreaming about. Now, the really interesting thing is electron microscope pictures of the brain, they're dry scans. That's not living tissue. That's tissue that has been dried out and plastinated and suffused with osmium tetroxide. But we need to figure out the strength of these connections. And I have always been doing a mild hand wave and saying, yeah, maybe we can figure it out from those pictures. But it's always been a rather limp hand wave because I couldn't bring myself to believe that was actually doable. Turns out that it's surprisingly doable. Not perfectly, but this is early days. So the Eckstein paper, they demonstrate that we get about 90% precision in actually identifying which neurotransmitters go in what synapse from this dry data. This is really amazing. That's a gift that I didn't expect. Then comes an even more fun paper. So Shio et al. They did the most stupid computational model you can imagine of a fruit fly brain using leak integrate and fire neurons. That's about as simple as you can go and still have a neuron that have a spike. Using this kind of inferred synaptic sign and connectivity. And they got some behavior out of this model. Stimulate some taste receptors and some motor neurons controlling the proboscis on the right side activate as if it would have moved the proboscis towards the source of sweetness. There are some neurons that make the organism twirl, and there are counterparts. This is even more bizarre that you actually can get this much done with that primitive model. Heaven knows what happens if we use a better model and actually can infer this better. So this is existing data and is probably hopefully going to get a lot of people working more on it. People are also doing some really interesting work going from the other direction. Suppose you have a body model. Nematode worms are fairly easy to model. There is also a lovely paper on fruit fly and behavior, which I didn't include because I was stupid and tired. And there is another paper here on a rat model. And in these cases, they're also using a top-down approach. Let's use reinforcement learning to make controllers that make these organism models behave, and then try to see, can we get then see if there is some similarity to the actual neural activity. This might be rather necessary for tying together body models with the neural models we're getting from scanning brains. There are some people saying that maybe we should actually just look at behavior and infer the entire brain connectome from that. I have my doubts that's doable. But given how much crazy stuff is happening with machine learning these days, let's try it and see what happens. I think there is something very cool happening. So to sum up, what are the timelines here? Right now, it looks like we have computer power is doing fine. The AI world and to some extent the cryptocurrency world have suffused the world with the GPUs. So it looks like we are not going to run out of computing power. The scanning modalities look rather good. At least the ones I mentioned, there are many interesting tricks here and people seem to be very, very creative on that side. Now, that seems to be then hindered, of course, by the cost of scaling up the connectomics. There was a report from the Wellcome Trust arguing that it's going to cost tens of billions of dollars and take about 15 years to do a mouse brain scan. But a lot of that was the proofreading. And it turns out that the tricks for doing the proofreading, especially if you get to genetically modify the mouse beforehand to act like a rainbow mouse, but on a synaptic level, then you might actually bring it down to the cost of a few millions. So that might actually be speeding up a lot more. The really tricky part here, and that is the embarrassing thing for me as a former computational neuroscientist, is, okay, we might have a connectome, we might even have data about what synapses there are and what transmitters and modulators there are, but we still need a lot of parameters, and automatically taking that input data into a model that actually runs well is still a lot of ad hoc work. And this is probably where the really interesting development has to happen if we are going to get to brain emulation. And this is where, imagine that the big intersection is. We basically need to set up a research loop. We need to have a pipeline from biological tissue to a model to testing that this model works well. That's going to require an interesting set of ways of testing against gold standards in biology. And then, of course, given that that's going to be wrong, you're going to have another system where you're trying to correct your model. You might need new modalities, new way of inferring relevant data. And this can probably be done with AI. We can't just rely on endless amount of grad students doing that work, because in that case, it's going to take absolutely forever. We also have seen a lot of really nice surprises the last 15 years. But I think being a gloomy Scandinavian, I'm also expecting that there are going to be some bad surprises too. So generally, the probability distribution is extremely wide. That diagram I got there is from a paper that's a few years older. Now I believe shorter timelines for brain emulation. Still, it's going. If it's done within 10 years, I will be very surprised. I think this is still a relatively slow process. So, to get to a punchline. So compared to most things that we're talking about here, this has a very, very low technology readiness level. We still haven't gotten the nematode worm to work really well. But it seems that it has a lot of interesting potential, because once you actually get to the point where you can start scaling it up, it's probably going to be possible to scale it up relatively rapidly. We don't know how long the fuse is going to be before that. And it can do a lot of interesting things that you cannot do in biology. It does play very well with cryonics. So if you have cryopreserved brains, this might be an interesting way of getting data out of it. But most importantly, that pipeline or automated research, basically roboticized big neuroscience, sounds like that's going to be darn useful for a lot of other stuff, not just for brains. We're going to be able to use expansion microscopy to look at cells at scale. I don't know what in aging benefits from that, but it sounds like it's probably going to be a lot. There's going to be a lot of these mechanisms and libraries and tools that can be shared between them. And of course, figuring out neuroscience and just being able to run experiments on simulated brains for Alzheimer's and similar things is also going to be super useful. So that was my little speech. Thank you.
Thank you very much, Anna. We've got time for 2 questions. 2 gentlemen in front.
Hi, thank you for the talk. I find this very interesting, especially the part you touch at the end of emulating complete organism models. That's, I think, very, very promising as a concept. The annoying question is, if we're gonna do this for life extension, how does the personal identity continuation work at all here?
We could keep on doing that for a long while. Personally, I think personal identity doesn't exist. I think it's something we talk about, but it's not really a well-founded concept. Still, a lot of people care deeply about it and can go on quite strongly about it. So some people are going to say, no, this is not preserving personal identity, so it's not a good way of extending life at all. Others like me have a crazy view of personal identity and have no problem at all with it. I think many of the philosophical questions are probably going to be actually resolved once you get stuff done. We can actually scan a few philosophers and see if they change their mind in the computer.
You preserve the illusion at least.
Yeah.
Yeah, so actually building on that, you started showing this example with emulating an old computer chip. And the big thing here is that you can image this computer chip and you can emulate the architecture. But if you're running software on this chip, it's essentially based on our current understanding of physics, it's impossible to actually capture the software state of the chip, right? And I think the same goes for cells, that we're running software, and by building the architecture, you may get a lot of insights in terms of how things emerge and so forth, but you don't get the personal self. You don't get to
I
build
beg
the
to
story.
differ on that, and there is one piece of empirical evidence for that, and that is people who have almost drowned in very cold lakes. They can literally be without heartbeat for hours, but they can recover with the right treatment. And the interesting thing is neurons can't fire below 26 degrees. So there is zero activity in the nervous system, yet they wake up and they are the same person. They have some amnesia because you also get blanking out of the intermediate term potentiation in the hippocampus and similar places. So I think that demonstrates that in some sense, if you could scan that hardware that corresponds to the cooled down brain, then it could boot up to the same person. Now, of course, if you don't have the right synaptic connectivity, you're going to get a different person or no person at all. So there is still a resolution here. And one important question is, can you actually grab all that synaptic connectivity using a scanning method? I think that's going to be the main problem.
One quick question from the back, and then we have to move on.
Do you have a cost estimate for how much it would cost to do the cryo-EM of an entire human brain? I talked with one of the co-authors of one of the Drosophila papers at some point, and we estimated it would be like $1 trillion for the human brain. Do you have any cost estimates?
At present cost, maybe it's probably a ridiculous number, but that's not going to remain at that ridiculous level. For example, there was that factor of 1,000 reduction if we can make proofreading better. That seems to be kind of, okay, that's one technical issue. There is also the scaling. Buying one electron microscope is expensive. Buying 10 of them is not 10 times as expensive. So there is an issue here about how does the cost scale as you scale it up. I personally think that scanning a human brain right now would be at least in the tens of billions of dollars, probably way more. But in 10 years' time, especially if we have demonstrated this for smaller brains, I think the technology would make it way cheaper. So I think in the end, my guesstimate is when it actually happens, if it happens, in that scenario, I would guess it's a multi-billion-dollar project. But it's just a multi-billion-dollar project. It's not a trillion-dollar project. But this is a guesstimate, and a lot of it depends on learning curves and how the scalability play out.
Right, thanks.
Great. Thank you very much.
Thank you.